it would be the largest reference collection of metagenomes, a field guide to the microbial universe, right? It's very parallel. Used by scientists for decades, well, maybe weeks until the next one comes along. Okay? But it's not a completely parallel story. Okay? And here's what's sort of different about it, and why this is really a project that's in tune with, I think, where the community is at. So first of all, unlike the Sloan, we have one telescope mapping the sky night after night after night, EMP will have distributed sampling. Thousands, hopefully thousands of scientists will be incorporating this into their normal work and additional thinking that they're doing about sampling. We'll have distributed sequencing. There won't be just one place producing sequencing. We'll have many places doing analysis. Where it's similar is that we'll need to have common protocols. Just like the Sloan project had common workflows. Once they got all the data coming in, they applied the same algorithms to it so they could reach comparable conclusions from different parts of the sky they were looking at. We have to do the same thing. We have to have common protocols for sample prep, DNA extraction, and so forth, and sequencing. And we have to have common standards, metadata, and standards about how we're going to manage the data, process the data, such that the project at the end of the day looks like one coherent collection as opposed to thousands of separate little projects. And there's some things we don't know about. Yesterday we were talking at lunch about, gee, maybe there should be a centralized archive of data. Just so that, I mean, there'll be many copies, but maybe there should be at least one copy that we protect, for example, like copying in many places. Maybe that's how we do it. Maybe we need to have a place where people can send samples such that 5 years from now, when the technology gets a lot better, one could reanalyze or resequence the same material, right? Maybe, we don't know. But so the point is that there's many things that have to be decided here, but fundamentally this is designed to be a community project that uses the magic of sort of networking in many ways, right, to act and function like a single global coherent project. So the model that we have in mind, and this I hope will be discussed at this meeting and laid out, is we would have a framework of some standard practices that enable us ultimately to compare data sets across many independent projects. And we have an organizational model that would target a handful of questions such that studies are get coordinated that really attempt to illuminate those questions. And this could be done by various coordination mechanisms, but where there's at least some effort to focus so that we can take those questions and say maybe fundraise against those questions to build infrastructure. Now, if you look at what's typically done in a field study, here's one I'm not involved in this one, I'm just getting pictures from their website to illustrate it. Some work that was done in sampling hot springs in Nevada. And here's a typical, at least for me, I'm a computer person, but for me this seems like a way that people think about this. There's all these red dots on this map. This is Nevada. Many of you probably know Nevada. And these are all locations of hot springs, hundreds of them. And they picked a set, maybe 50 or so, to sample. And they went in little teams and they collected material, trying to do it the same way, and put it in freezers, and then they were, of course, doing observations against, screening it, and some of it was being sequenced and so forth. But this was a typical sort of fairly large-scale effort. And of course, in our mind, we're talking about hundreds or thousands or more kinds of studies like this that would be attacking different questions around different parts of the world. So one thing we could do, for example, is build an infrastructure for coordination that allows people to articulate what field studies they're going to do. Where are they going to sample? What have they already sampled? What were the questions they were asking? What did they find out about that environment? Did they measure physical parameters about the place where they took the samples? When did they take the samples? And so forth. And put this in so we have metadata that's comprehensive, but we also can get a global picture of many independent projects and see where there's holes. Maybe no one is deciding to sample a certain class of environments, and somebody then could see that and go and do that. Okay? So an infrastructure for coordination is really important. There's been a lot of discussion, this is not, I can't be authoritative on this because I don't do wet lab things, but it's clear we need strategies and standards for sampling, methods that have to be tailored to the specific environment, whether it's soil or marine systems or whatever. We need to have georeference metadata so we can map it in space and time. We have physical parameters. We need some kind of standardized way to do DNA extraction, maybe several, depending on the types of environments coming from. We need a set of protocols for sequencing that are maximally comparable to each other so that different centers can hopefully get the same result if they do the same thing. And we have to do some of this for 16S and some of it for whole genomes. And we need to define a set of standardized analysis pipelines, building on what the community is already using. And the way that we think about this is to begin with defined and totally open protocols, and they'll be imperfect at the beginning, but they'll be associated with the EMP, and we'll have some process in place that will allow people to improve those and push them back out. And of course, do that in some kind of controlled fashion so that we always know what it is the most recent version. And allow groups to do research to improve the protocols by showing that some new protocol, not only is it better than the existing ones, but is more robust. Now, why do we need this? Why is this project even sort of contentedly, interesting or feasible? Well, as far as we know, microbial life on Earth is vast. Maybe 10 to the 30th organisms, which, if you believe the astronomers, is about 10 to the 9th times bigger than the universe. So we're attacking something that's 1 billion times bigger than what the Sloan was attacking. We really don't know how many species, or even what a microbial species is really, but we know that there's a lot of diversity out there, and there's massive gene and protein diversity, and getting a handle on that, even a glimpse of whether we're closer to the lower limit or the upper limit would be really useful. Sort of articulated, some of these things. We imagine that this could be organized in themes with different PIs pushing different sub-projects, but again, doing it in a framework that allows maximum comparability and leveraging the existing community structures. So we've been thinking about things that could be done as pilots. Some of these have been proposed to existing sequencing opportunities. Some large-scale surveys, a small number of some very deep demonstrations showing that we can maybe sequence to extinction the diversity in single locations. And a variety of hypothesis-driven sort of problems related to questions that might be of interest to different funding agencies. And I'll walk through maybe one or two of those in a second. But there's also a lot of work that has to be done to technically debug whether this concept actually makes sense. Can we come up with standard protocols and then use them and actually have more than one center using the same protocol and get enough quality in the data that it works, right? Some of the questions that we have to do is, can we do very high levels of multiplex sequencing for very broad, relatively shallow surveys, right? Given the throughput of the current sequencing machines, in some cases we don't want to throw a terabase at a single sample. We want to throw maybe a very small amount, or not small, but moderate amount of reads at a single sample, which means it implies very high degrees of multiplexing. Can we characterize the parameters in an environment in a repeatable way such that you can correlate them with what you come out of the sequencing? Can we get the community, or can we build mechanisms for collecting metadata and maintain this in a sample database such that the community actually uses it? And can we get analysis pipelines out there and teach people how to use them and so on? So there's both science targets, but a lot of technical work that has to be done to do this. And if we imagine going after funding for the project itself, a large amount of that resource would have to go in making the technical feasibility of the project. There's many, many basic science questions. I'm not going to go through these, and I've mentioned some of them already, but basically there's almost no problem in environmental microbiology that this project wouldn't, in some sense, inform or improve on. Some of these are very controversial questions at some level about how cosmopolitan microbial taxa are, for example, or whether we really have biogeography. There's a lot been written about the current sort of poor state of our understanding of microbial diversity. This is a paper back from 2004, Curtis and Sloan, that I sort of like as a way of framing this. They're trying to relate global microbial diversity to why we should care about the global diversity in the context of local communities, and whether these patterns could have an impact on studies that try to link community function, structure, and strategies to really abide outcomes, like figuring out whether there are certain places, for example, that would be more fruitful for looking for microbes that might be relevant for drugs, for example, as opposed to point A versus point B, or for identifying studies for finding novel chemicals. And one of the key observations here is that is this notion that to understand how a microbial system works, you've got to understand the community from which that system is actually constructed, and try to pull apart those factors that are random from those factors that are deterministic. And a study like EMP will collect enough data where you could finally start to see some of these issues come to bear. There's a lot of interest in going very deep in some places. This was a paper from many years ago, 2002, relating to trying to estimate global diversity. This is where this notion that the global sort of taxodiversity is somewhere between, say, 1 million and 1 billion by using some of these simple estimators. There's questions about what is the shape of communities, and what are all these rare taxa doing? Are most of them asleep, or are they inactive, metabolically inactive? And do microbial communities fundamentally function differently than macroscopic communities with all this sort of passive diversity hanging out? Does that really change how microecology functions? There's reviews of microbial biogeography, and there's sufficient evidence, I think, that's emerging that microbial biogeography probably does exist, as the paper that Ted was on. But if it does, can we map it? And can we map it in some reasonable way, in some cost-effective way? That's sort of the macro question here. This question about global dispersion versus endemism in microbial